We know that soil fungi that associate with plant roots can be really effective for drawing carbon dioxide from the atmosphere and storing it in soil. But we don't know, one, exactly how they do it, and two, how much carbon they can store. What the research that we're doing here at Lawrence Livermore is trying to help us understand is, one, the specific mechanisms by which root-associated fungi transfer carbon from the root into different soil carbon pools, and two, how much they can do that and how persistent the carbon that formed is over time.
1 gram, or let's say a teaspoon of soil, can hold up to 1 billion cells of microorganisms. But actually, only 5% of those microbes are alive on any given day. Most of them are actually dead, and we sometimes call that necromass. It's just sort of the leftover husks or shells of fungi and bacteria and even viruses. And these are organisms that were living when the plant grew, but maybe they're decaying at this point. But they've got all sorts of different molecules that are going to stick to mineral surfaces, perhaps get trapped in little tucked away, out of the way spaces in the soil. And that carbon over time, if it's far away from an organism that might want to break it down, will start to accumulate. We've built a one-of-a-kind isotope labeling system for plants. There's really no other place on the planet where we can do high replication studies of how carbon is flowing through plants and into soil using 13C carbon, which is a stable but rare isotope of carbon. So that's really kind of the special sauce of this particular facility, is that ability to track carbon through pools that would be otherwise difficult to measure.
The collaboration with Lawrence Livermore National Laboratories and UC Berkeley was really exciting. UC Berkeley had a unique setup for tracing carbon from the air through the plants into the soil. And Lawrence Livermore's advanced methods enable us to trace that carbon all the way down to the DNA to differentiate which microorganisms in the soil have the carbon from the air.
We have automated stable isotope probing so that we can do high throughput analysis of many different samples. So this is a process where it used to full-time take a person a week to do 6 samples. And now we can do up to 24 or 32 samples. So it's really been able to enable high throughput studies of looking at soil microbiome function.
The goal of this collaboration is to really understand the mechanisms by which Loam's fungal isolates are drawing down carbon through the plant and storing it in soil. And the way we're tracking the flow of this 13C carbon is by using some of Livermore's cutting-edge imaging technologies like the nanosims, so we can really visualize how carbon is being sequestered at the micron scale.
I love this project. It's really exciting. I'm really interested in the possibility of using soils to draw down carbon globally. And so I'm really interested in what our technology can do to bring new insights into soil processes. These are really difficult to study. They're opaque, they're underground, they're very micro scale. So you have to separate out processes that are occurring at a really small scale to understand what's going on at a global scale. What we bring with the imaging capability is the ability to actually directly observe the transfer by tracing these isotopes. The instrumentation was essentially developed for cosmic chemistry sorts of applications. For example, you can find that there's a piece of star that exploded in a distant location that had been incorporated into a meteorite that we find here on Earth. That's the sort of technology that we're using for this. So it's really the fact that we are able to combine these advanced genomic tools with basically a biogeochemistry physics type of tool that makes the collaboration unique.
Soils are incredibly dynamic and alive ecosystems that contain millions of different organisms in tiny volumes, like a single gram of soil. And I'm really motivated to understand all the ecological interactions that occur between fungi, bacteria, different animals and soil, and how those play out over tiny scales of space in the soil, like the scale of an individual mineral grain. Also, if we can better understand those interactions in which microbes are active and are drawing carbon down from the atmosphere, we can hopefully help accelerate the pace by which they pull down carbon from the atmosphere and slow the pace of climate change.